Ma'am, we'll start. Yeah. So good afternoon, everybody. So delighted to have everybody here for the second lecture in the M.S.Swaminathan Centenary series. In the first lecture, we had Professor Ronnie Kaufman, and one of the things that he mentioned, he's a plant breeding specialist, is that plant breeding in the next era will be driven by genomics and big data. And I think segueing very nicely from the talk that he delivered in the first, as the first in the series, we have Professor Rajeev Varshney, who will be delivering the second talk. And he will be talking about genomics for food and nutrition security, striving to realize the vision of Bharat Ratna, Dr. M.S. Swaminathan. I think Dr. Varshney doesn't need any introduction. He's a great collaborator, and he's currently the director at the Center for Crop and Food Innovation. He's also the director of the WA State Agricultural Biotechnology Center, International Chair of Agriculture and Food Security, and the Food Futures Institute at Murdoch University, Australia. He wears many hats, and he's also been involved very extensively at ICRISAT in developing genomic resources not only for chickpea, many of which of those varieties are now been released recently, but he's also been involved in a lot of international and national collaborations which have paved the way for many of the genomics of the crops that ICRISAT had been working on. So without any more delay, I would like to hand over the podium to Professor Varshney. Thank you very much for agreeing to give this talk. So you can take over now, sir.
Thank you very much, Madam. Hope you can hear me. And you can see my slides as well.
Yes. Thank you.
Okay,
yeah.
good.
You may begin. Yeah.
Thank you very much. Let me thank you once again for providing me this opportunity. And it's really an honor for me to be the part of this prestigious series, the M.S.Swaminathan Centenary Lecture Series. And I'm grateful to Dr. Swaminathan Madam and also other Swaminathan Research Foundation leadership for providing me this opportunity. So in this presentation, I would like to share some of my views on genomics for food and nutrition security, but also would like to pay my tributes to Professor Swaminathan. In fact, when I will present, you will realize that some of the work what we are doing, this is towards realizing the vision of him. And in this direction, as we all know that we had a big loss of losing Professor Swaminathan last year, but at the same time many of us, we celebrated his legacy, his work, and the whole world. Though we are missing him, but he has left a legacy, and all of us are trying to take this legacy forward. And though I paid my tributes in the different articles, in the case you have not read, then you are most welcome to see on my blog or even the Nature India. And not only that, even the National Academy of Agriculture Sciences, and also in the Review of Agrarian Studies, where we contributed, we shared some of my experience while receiving the mentoring and then supervision or inspiration from Professor Swaminathan. Very briefly, I would like to mention it especially to our colleagues, young colleagues, because Professor Swaminathan has been an inspiration to all of us. For me, I met him first time in 99, and this picture when I was very young boy and was doing the PhD, I came to SRF in 1999, where Dr. Lalji Singh at that time point, in collaboration with Professor Swaminathan, organized a conference of AdNet. And I remember that I received the best poster award there, and I received that an envelope with the 2,000 rupees, and this I got it from the hands of Professor Swaminathan. This was really a big moment for me in my life. After that, I met him in 2000 in the second International Congress in Crop Science Congress in Germany. And while I was in Germany for my postdoc from 2001 to 2005, I stayed in touch with him through letters and emails. But my interaction started with him when I joined ICRISAT in 2005, and I had a privilege of interacting with him more frequently. We had several occasions, and I feel really privileged to visit MSSRF many times, and also inviting him at ICRISAT. So, and this you can see several pictures. And not only me, he was very close to my family as well. And whenever he was coming to ICRISAT, we were always inviting him for a cup of tea or so. So this is really good. One thing also I would like to mention, because as you know that I have been working in the area of genomics. So whenever he was asking me about the genomics, and I say, sir, I'm working on this or this, he will be always listening very patiently and will be asking different kind of question. What does it mean or so? And whenever he comes to ICRISAT, we used to take him to the lab, explaining that what is happening. And this was really wonderful and you can see a person like him always listen these things. And why I'm telling all these things? Because when he was coming and he was asking all these questions about the genome sequencing, in one meeting, I think this was in 2013 or 14 when he came there, and I told, can I have your consent to sequence your own genome? And he laughed. What does it mean? I said, well, I tried to convince him. But thing which I want to tell, that on his 19th birthday in 2015, I had a privilege to present the genome sequence of Professor Swaminathan to him. And after posing numerous questions about his genome, he asked me, or he reflected, and this was really very inspiring. He says, when I was working on my PhD, I began to understand the double helical structure of DNA, but I never imagined I would one day hold the fully decoded sequence of my own genome in my hand. So such moments and interactions have served as a beacon of inspiration for countless young minds in agricultural sciences, both in India and internationally. Another incident I would like to share, and this is in 2016, when he was there for one of the meeting of FAO as a keynote speaker, and I was also there. And I remember that Parasraman from MSSRF, he was there, and at that time Sarol was already in the wheelchair. So I asked him while having a cup of coffee with him, sir, you have already contributed so much to international agriculture. Perhaps you should not restrain yourself by traveling so extensively. But before I could finish, he replied, Rajeev, interacting with young minds like yours at scientific meetings keeps me abreast of the latest advancements in agricultural science. That's what motivates and energizes me to travel at this age. I said, wow. And he added, I can't imagine just sitting in my office when I believe I can still make a contribution to society. So this was the way that Professor Swaminathan was working. And I fondly remember his last email, which I received from him in May 2023, when I received that fellowship of the Royal Society. And as you can read that message, he says, I'm very happy that you have been elected as a fellow of that Royal Society, and so kindly accept my congratulations and very best wishes on your fully deserved accomplishment. So I always feel very inspired whenever I receive an email. I'm not sure whether Dr. Madhura Swaminathan is there or not, but I would also like to highlight her email, and this is especially this sentence when she mentioned, my father is happy and has already written to you. I believe while talking to me today, he said, a fellow is someone who makes an original contribution. So these are the ways that I have been inspired by him. And when we are talking to Royal Society, I would also like to share another event or incident. In Royal Society, they have a charter book, and this charter book was created in 1663 after King Charles II granted a second royal charter to the organization, establishing the structure of the Royal Society. And since the first signature was entered on its pages on 9th January 1665, this volume has recorded the signatures of new fellows and foreign members as they were elected every year. And last year when I went there, so Royal Society generally asked that, do you want to see some signature in particular? And as you can imagine, that I requested to see the signature, the very first signature of Professor Swaminathan, which was inscribed in 1973. And I was so excited, you can see with my smile, that wow, I found the signature of 1973 from Professor Swaminathan. And finding and beholding his signature filled me with elation. It was as if I was floating on cloud 9. Moreover, I felt privileged to sign the same book in 2023 in my 50th year. And now the same Royal Society, they have invited me to write a biographical memoir for Professor Swaminathan, and I'm working on this thing with Dr. Sharat Kumar and also Dr. R.S. Paroda. It's really privilege for me to contribute to this one, and I'm also thankful to MSSRF for providing us the permission and several pictures, and this memoir will be available very soon, and this will be in public domain. So this is really very nice, and I'm not sure that many of you might have seen this picture of Professor Swaminathan from Royal Society. This is one of those rare pictures, and I got it from Royal Society when he visited way back in 1973 or so. So these are some of those things that I wanted to share, especially with my young colleagues, that these kind of people, they have been inspiring that whole generation and many of us to contribute towards the food security and nutrition security, some of the key things which have been very close to his heart. And right now also we have lots of challenges for food and agriculture, and this is not that these challenges are new. We had, in fact, many of us, or at least I have read in the books, and many of senior colleagues, they know that sometimes way back in 1960s, in the context of India, we had really serious challenge of food. There was a book published by Dr. Paul Ehrlich in 1968 titled The Population Bomb. Even on this book, in the cover, you can see this highlighted in yellow color. This was written, while you are reading these words, 4 people will have died from starvation, and most of them children. So this is very discouraging or disappointing book or so. Even some of the paragraphs, and if you would like to read, the battle to feed all of the humanity is over. In the 1970s, hundreds of millions of people will starve to death in spite of any crash programs embarked upon now. At this late date, nothing can prevent a substantial increase in the world that rate. They wrote, I have yet to meet anyone familiar with the situation who thinks that India will be self-sufficient in food by 1971. I don't see how India could possibly feed 200 million more people by 1980s, because at that time the people were about 400 million people. But this was this time, but at the same time, and this was the time that Professor Swaminathan together with Professor Norman Borlaug, they were sowing the seeds of Green Revolution. And we all are aware, well aware of the success stories of the Green Revolution, where we had this Green Revolution cereals through the transformational genetics in the form of dwarfing genes, RST alleles, in the case of wheat, as well as in rice from the Dr. Yon Longping from China. So the world will start to have huge amount of wheat and rice, and salute to the Green Revolution heroes. Of course, Professor Swaminathan, Norman Borlaug, Yon Longping, and also Professor Gurdev Ghosh, that they contributed a lot in this direction. And as a result, if you would like to see in the context of India, from 1961 or so, now if you see, this is the old data 2018, but things keep on increasing. If you see the cereal crop, cereal production has been increased more than 200 or 300%, and cereal yield has increased. So even population is increasing with this rate, but that grain production has been increasing with much more faster pace with the limited land use. So you can see that land use has not increased heavily, and so we were able to produce more cereal crops. Now India, where Paul Ehrlich wrote those kind of disappointing words, now you can see that India has become a food prosperous country, or from a grain deficient nation to food security for all. Now India got 1.4 billion people, more than 3 times than what we had in 1960. But currently India harvests around 110 million. Now this year there will be going 110 million tons or so, and is one of the lead exporter in the wheat. Even those calories, if you would like to say, the per capita net availability has increased to about 312 grams per day from 312 grams per day in 1950s to 512 grams. So though we still have some issues of those malnutrition and food security, but good news is that Ehrlich's prediction about famines and massive deaths were found to be false. And Jonathan Last in 2013 in the New York Times, he wrote The Population Bomb probably was one of the most spectacularly foolish books ever published. And he wrote this thing in his book as also in that other things. So I think these are that, how this could happen just because of the contribution of Professor Swaminathan. Today, the world can produce almost 3 times, and I was talking earlier about India, now I'm talking about the world. Almost 3 times as much as cereal from a given area of land than it did in 1961. Yield outputs have increased. More than 241%. Cropping intensity has been increased. Lot of infrastructure, especially for irrigation facilities, etc., have been increased, and improved farming incomes, mechanization. But at the same time, we also had that fertilizer usage as well. So all these things happened, and now if you would like to see the whole world map, and you would like to see the yield, countries like India, China, Europe, North America, or in Australia, where we are having this grain belt, you can see very high yield, green color thing, yellow color thing. There are still some regions left in Africa and some other places where they still have that huge potential to enhance the yield. So these were some of the success stories, and this is, we can feel very proud of that agriculture, because many times agriculture is not appreciated. On the other hand, human health, which is important, and both things are important, is appreciated much more than agriculture. But this is the way that you could see that when people were predicting, but all those predictions were false because we were having enough food. If you would like to see the current scenario, we know that by 2050 we will be having 10 billion population, and now if you will like to see, even at this stage, 1 in 10 people do not get enough to eat. Hunger is also on the rise in the Western Asia as well. There are conflict and climate crisis, and there are several places on the earth they are still very hungry. So I mean, this is not that we have solved all the problems. So this is not very good situation. And in terms of the nutrition, nowadays we are focusing a lot, and many of us, including WHO, FAO, in Sustainable Development Goal, everywhere we are talking about the nutrition, which is very important. But, and this is much more important in ongoing conflicts and geopolitical issues worldwide. So I think that these are very important issues. Climate change is posing another threat, and all these issues, they are basically putting scientific community that we need to produce more food, and we need to contribute toward the food and nutrition security. These are the things that are becoming important now. But if you would like to see again from Professor Swaminathan, in 1986, he wrote this book called Global Aspects of the Food Production. Also, he gave a talk in FAO at that time point, and there are some excerpts from the 2017 from Chennai. And he also wrote one article in Indian Journal of Medical Research in 2013, where he highlighted that food and nutrition security, they are intermittent for that word, and since only a food-based approach can help in overcoming malnutrition. So he was so visionary that he was looking all these things much more ahead than many of us can think. And so in 2017, he stressed the need for a change in the emphasis from food security to nutrition security. Of course, this food security, nutrition security is a result of several things. So not just crop improvement, but policies and a lot of other stuff. But now people like us who are working in agriculture space, in crop improvement space, we need to think in this direction. And what we are thinking nowadays, that we need to have the sustainable agri-food system. Why? During last few years, 40 to 50 years, we know the temperature is increasing during the climate change. Greenhouse gases emission is also going higher, population also increasing. Now what we need to do, that to feed this ever-growing population, we need to do several things. We need to bring this temperature down. We need to bring this carbon dioxide gases down. At the same time, we need to produce the crops which are climate resilient, which can give more food, which can provide more nutrition, and not only the crops, but we are talking about agri-food system where the trees, fisheries, and a lot of other stuff comes. So we need to work in this direction, that how we can contribute toward the agri-food system. And this is some of this work of Dr. Swaminathan that he has inspired me, and I have been working for last 20 years in this space. in different capacities in the different organization. What is required, what we are doing, and I will share some examples. Some work from ICRISAT, some work what we are doing now at Murdoch University. So what is happening that if we need to develop these crop varieties which are climate resilient, nutritious, with higher productivity, sustainable, we need to integrate genomics in the breeding program. We call it this genomics assisted breeding. Then only we will be able to address or to achieve this at least 3 SDGs: zero hunger, good health and well-being, and climate action. What is this genomics assisted breeding? When I was in Germany, together with Professor Andreas Granat and Professor Mark Sorrells, we gave a concept of integration of genomics in breeding, and we called it genomics assisted breeding. This was in 2005. This was the time that genomics was just booming up, and then we had the first genome sequence of the rice crop. So we provided a concept in the 10th anniversary issue in the Trends in Plant Science, feeding the world plant biotechnology milestone, that what we can do. I'm so happy to mention that during last around 20 years or so, there are lots of advances in the genomics assisted breeding, and there are lots of success stories. But for doing this thing, first, you need to understand the genomes. You need to understand the traits so that you can work, you can define the appropriate combination of alleles, etc. In this direction, when I was working at ICRISAT on the crops like dryland crops, pigeon pea, chickpea, pulse millet, peanuts or so, together with several ICR institutes, state agricultural universities in India, many organizations from all around the world, in China, in Europe, Australia, North America, we developed several international consortiums. And then we led some of those consortium, we decoded the genomes of pigeon pea, chickpea, pulse millet. So genomes basically that you understand the entire gene repertoire, and you are having all these genes in your hands. And not only for the dryland crops, many other crops which are important in Asia like sesame, mung bean, ajuki bean, pea, etc. So we have been working on all these things. But does this mean that if you find like in 28,000 genes are present in the case of chickpea, whether all 28,000 genes are expressed in all the tissues at all the time? Probably not. So we need to understand their gene expression in temporal and spatial manner. And for that, we developed the gene expression atlases for chickpea, pigeon pea, and groundnut. This is one thing. Second is that initially when we were developing the genome assemblies, they were based on the short sequencing reads. But with the time, we kept on advancing the genomics technologies. And during last few years, we have been developing these high quality genomes. So in this paper, the same legume genomes what we presented earlier, we brought them at the chromosome level and we call them pseudo molecule. And we got the chromosome length genome assemblies as well. This is an example in the case of pulse millet, for instance, that our earlier genomes, they were very, like for instance, this was the TIFF 2017, but then after that we developed this platinum standard reference genome. Now you can see that we have improved these genomes very high level. We have addressed lots of issues. So you can do a lot of work. And now even in the case of soybean, we have developed the genomes and we call them gapless genome assemblies. We developed this thing for the 2 soybean cultivars also in collaboration with United States and Hong Kong. And one of my colleagues, Vanka Garg, so she did really good work on this aspect. So we have been able to assemble those genomes now, gapless genome, and we can get, and I will talk about later, telomere to telomere genome assembly as well. So this is one way that you can understand the genes. But now if you would like to use them for the breeding with that objective of the food and nutrition security, the next thing is that we need to understand the genetic variation. And for that, there are large scale germplasm available in different gene bank, either this is in India, in NBPGR, ICRISAT or CGR gene banks, or here in Australia, Australian grain gene banks. You got huge germplasm collection. And we all know that during the case of this, now when we talk about the breeding, we had the domestication, we had the breeding. So because of these two bottlenecks, we lost many favorable alleles. And now what we need to do, we need to go back to the land races, we need to go back to the wild species, and we need to understand their genomes, and we need to use those genes which we have lost. And based on these things, there is a concept of the pangenome where people were talking at that species level. We moved one step further and we say when we want to target the wild species, we should not just talk about pangenome, we should talk about super pangenome at the genus level. So because of these kind of concepts, we started to do these pangenomes and super pangenomes. Like in the case of chickpea, we got 8 different wild species. So what we did, we assembled the genome assemblies for 8 different wild species. So not one genome, 8 different genomes, and we recently published this thing. And here we have provided the sizer super pangenomes. We got an idea, information on the genes present in different wild species. And we had many colleagues involved in this project from ICRISAT, from many Indian organizations, and many other organizations in Australia, China, and United States. Other one is in the land races. So now, for instance, in the case of pigeon pea, we targeted about 292 pigeon pea lines coming from 23 countries. In the case of chickpea, we targeted 429 chickpea lines. And then we just also did the phenotyping for the traits in which breeders are interested. And while combining these things, we got the genes associated with the different traits. Not only in this case of chickpea and pigeon pea, we did similar kind of work in the case of peanut in collaboration with Guangdong Academy of Agriculture Sciences this year. And here we also developed the genome variation map in the case of peanut. And we got that genetic association with more than 28 agronomic traits. We are doing similar kind of work in the case of pea with Chinese Academy of Agricultural Sciences, where we have developed the pangenome. In the case of azuki bean with Beijing University of Agriculture. So nowadays our lab, they are working on different crops, and idea is that how we can understand this genome architecture, etc. When I was at ICRISAT, we embarked on a big project, and this was the sequencing of more than 3,366 lines. This work was done in collaboration with ICRISAT, ICARDA, and many other organizations. And this was a huge work. And based on this thing, we developed a chickpea pangenome. We got several new genes, and then we extended this study further by sequencing more than 10,000 lines. This is the world's biggest project. Earlier, the 3001 was the big project, and right now this is the biggest project of sequencing of plant genomes, where we sequence more than 10,225 lines from all over the world, and not just for fun, but to understand the genes associated with different traits. Now we are going not only for the genes, we are identifying the haplotypes, we are developing the haplotype maps, we are getting the hot spots where we can get more genes, and this is really huge information and very useful resource for the breeding. Next thing is that so far people have been developing those genome assemblies. They already moved to that chromosome scale level, but they are not reaching to the full chromosome, which is called a telomere to telomere. So recently we have provided this concept that why we need to have the telomere to telomere genome assemblies, so that we can unlock the full potential of plant genetics for the crop improvement. And inspired by this work, now we are the partner in several international genome consortiums, not for one genome, but larger genomes, like PISM 50 plus genome sequencing consortium being led by Canada from Murdoch University. We are the partner there, and we are contributing towards sequencing of these genomes, where we are having 50 plus, and each genome will be that de novo genome assembly at telomere to telomere kind of thing. And from Australia, we are contributing 3, but 2 other excisions we are sequencing from Mexico and Afghanistan. Another consortium, we are the part of that one, International fava bean pangenome consortium. Fava bean is a big genome, and here again this is being led by IPK, and also now one of my colleague Muru, who is moved to the Texas. So those colleagues, they are leading this consortium. We are the partner there. We are also contributing several genomes in that even fava bean. Not only that, here in Australia also, we are targeting 15 Australian chickpea varieties. Chickpea is very important crop in India and also here in Australia. And now we are doing this assembling of those 15 different genomes. So this will be also a pangenome of Australian chickpeas, and there are a lot of stuff whether we can do it. I would also like to highlight another big project, and would like to invite that in the case if some colleagues are interested, we will be happy to work then. Together with IPK and Helmholtz University of Munich, We are leading international initiative on development of wheat pangenome and haplotype catalog. We had several meetings in 2023, 24, and even in September 2024. Plan is to sequence or to assemble the wheat pangenome for 100 different wheat varieties from different countries. And many countries are the partner there. And we are looking for more countries which are missing here. And in Australian perspective, we already have targeted about 15 wheat varieties which are coming from the different states. We are working in collaboration with different partners, and a lot of sequencing work has already started, and now genome assembly is underway. So this is the way of the genomics, that how and what you can develop now, decode. Earlier we had the tough time to even sequence one genome. Right now we are talking to T genome, and we are going in thousands and tens of thousands. We have not reached yet in that range of the hundreds of thousands, but that day will also not be very far, where we should be able to sequence, or where we will be talking in sequencing hundreds of thousands of the genomes. Anyway, this is one part. Second is, we need to translate this research. We need to, again, our goal is how we can contribute towards food and nutrition security. For that, we need to understand, we need to associate those genes with the phenotypes. And many countries, many places, phenotyping is still a bottleneck, because you need to understand, you need to evaluate your germplasm in that glass houses, greenhouses, field conditions, etc. And you need to collect that phenotyping data. In some cases, we collected the phenotyping data, we associated with that genomic data. And now in the case of chickpea, groundnut and pigeon pea, we were able to map more than 20 to 50 traits like drought, heat, salinity, ascochyta. In the same thing in the case of groundnut, late leaf spot, leaf rust, iron deficiency in the case of pigeon pea also. So we have been able to deliver that molecular markers associated with the traits in large number of crops. And this work again in collaboration with Indian Institute of Pulse Research, Directorate of Groundnut Research, many state agriculture university, IRI, many other organizations in India, because these were the colleagues who did a lot of hard work. They did a lot of phenotyping, collaboration, and of course outside India as well. So this is based on the collaboration and partnership. This is not work of just one person or one institute, but multidisciplinary, multi-institutional contribution. Once you got those genes in your hand, next thing is how to mobilize them, how to bring them in the new genetic background. And for that, we had these discussions way back in Nature Biotechnology in 2012, like MABC, MARS, they are the different approaches. But when we did the discussion that what is the challenges, and we realized several challenges. One is that if you would like to use them in the breeding program, one, the genotyping cost needs to be cheaper so that breeding programs can afford that genotyping. Second, our breeders need to be skilled at how to integrate those things, so they also need to be trained. Third, that we need to have this support from the public policy or from the government, because in some of these crops, when we started chickpea or pigeon pea, even at that ACIR level, we did not have that accurate trials, that guidelines for testing molecular breeding products. So, and we worked at that time point, Dr. Shopan Datta, he was the DDG Crop Science. We worked with his office. We worked at that time point, Dr. Mohapatra, also in the Director General of ICR. So we did a lot of work with the directors of the institute, Dr. Radhakrishnan from DGR, Dr. N.P. Singh from IIPR, and of course, Dr. Bhardwaj, Dr. G.P. Dixit. There are many colleagues from India. We worked with them. So anyway, we tried to address each of these three different components. What we did, together with the Gates Foundation, I led one project, we call it High Throughput Genotyping Project, and the purpose of this project was to bring the genotyping cost lower. Through these innovative approaches and with that economy of the scale, we were able to bring the genotyping cost for up to 10 markers. The cost was $1.5 per sample, including the DNA extraction cost. And I feel so proud to lead this project that now this project is being, or has this legacy, and now 8 different CG center, 13 years program for private companies, and they're using this project outputs in 28 countries across 18 crops for more than 100 traits or so. So this is the way that this project brought the genotyping cost down. Second, together with my colleagues, especially here, I would like to mention Anu Chitkineni, who was that manager of that Center of Excellence Genomics and Systems Biology at ICRISAT. She worked with me, and together with her and many other colleagues at ICRISAT, we organized 15 different training courses where we trained more than 485 breeders, not in the data generation, but in the integration of the genomics in the breeding program. From India alone, we trained more than 365 breeders. From Eastern Southern Africa, 49, Western Central Africa, 40, and other countries, 31. So this was a huge work during the last 10 years or so. And as I said, we also worked in the terms of those guidelines or so. And then those lines, what we did, this molecular breeding lines, they were tested. So like breeders, they did this introgression work. We provided the genotyping support. They did this evaluation across the different environment or so, and following all the guidelines. Many of these lines were released as a commercial or as a varieties in many countries. And here, this is the success story in the case of chickpea. And here, many of you already know from India, Dr. Bharat, very leading chickpea breeder from IRI. And together with him and many other breeders from Indian Institute of Pulses Research and in fact from IRI as well, we were able to deliver several chickpea varieties which are coming through the genomics assisted breeding. Now you can see the yield. Generally, you are having 1 to 1.5 tons per hectare, and you got the varieties more than 2 tons, 2.5 tons, 1.9 tons per hectare, etc. They are for the drought tolerant. Same thing in the case of Fijian wilt, coming like Pusa Manav, and here is coming from the University of Agriculture Sciences, Gulbarga, and many other organizations. So we kept on delivering these kind of varieties. Now, many of these varieties, they are already grown by the farmers. In the case of ASCO catabolite, from Punjab Agriculture Universities, etc. Not only in India, in Ethiopia also. Because we were leading this project from the Bill and Melinda Gates Foundation, and 2 varieties, Jele2 and Grar, they were released in Ethiopia. Like my breeders' friends, Niguse Girima, Asnake Fikre, they played a key role, and you can have these varieties up to 3.8 tons, 2.1 ton per hectare, and these were coming. And the drought is very complex trait, but we were able to deliver these varieties. Similarly, in the case of Kenya, for instance, several lines have been delivered. In the case of Kenya, in collaboration with Paul Kimurto. In the case of Tanzania, lots of stuff that has gone to the farmers' field, and those people, they are really working, or they are being grown by the different farmers, etc. In the case of peanut, in Karnataka state, we were able to release the variety foliar disease resistant. And this is the guy, Dr. Ramesh, what he did this great work. We had first set of that high oleic acid varieties, and lots of now these varieties in the case of Ghana, and Dr. Radhakrishnan played very important role in high oleic varieties, etc. So we got lots of varieties in the different crops, and this was the document from the FAO in the case of chickpea alone. And you can see that in the case of chickpea alone, more than a dozen varieties have been released in India, Ethiopia, Kenya, and Tanzania. Same case in the case of groundnut, in the same case in the case of pigeon pea, etc. So these are some success stories, how you will be able to deliver those genomics in the breeding program. I think somebody is doing some new sense that started to putting that screen. I don't know that Gayatri or somebody, you can address the issue, because they started to use the sketching on the screen. Anyway, so now coming back to some of that work, what we are doing here now, in addition to those legume work now at Murdoch University, we are working on wheat. Wheat is also very important crop for Australia. This is key crop, one of the leading crop. And what we are doing here, that in wheat, we are improving wheat for the nitrogen use efficiency, because right now farmers, they use huge fertilizers, and these fertilizers is not being used, or nitrogen is not efficiently used by the plants. So there are lots of leaching and then going soil pollution and creating that, in that contributing to the greenhouse gases. Now our idea is that how we can make that wheat nitrogen use efficient. Similarly, another project on the heat tolerance, how we can enhance the heat tolerance in the case of wheat. And we recently organized a conference, International Wheat Congress in Perth, where we brought more than 950 people from more than 52 countries. Now some summary is also coming in the Nature in 1 or 2 pages. So like this will be shared in public domain. But so this was a big story, success story, and we have discussed a lot with different breeding programs or so. that how these approaches can be moved further. In the case of legume genomics, we are working on that project on genetic gains. One of my colleagues, Rutvik Varmuk, is leading this project, and here we are working on 3 different crops: chickpea, fava bean, and lentil, and we are using this genetic diversity. We are working with different organizations. Another project is with the University of Queensland about the nitrogen fixation in the case of pulses crop. We are also working on other project, and this is enhancing the heat tolerance in the case of chickpea, drought tolerance in the case of chickpea. So we are working in range of the projects dealing with those climate change related issues, etc. The other thing, now my group is also working on the horticultural crop genomics, because not only just legume and cereal, horticulture crops, especially fruits and vegetables, they also play a very important role when you are talking about the nutrition security. And now here in Australia, we are developing advanced genomics platform for Australian horticulture. In another project, which is the RACE project, we are developing the cost-effective genotyping technology. And then recently, we are having another big initiative called Genetics for Next Generation Orchard. And this is led by QUT. We are also leading one of the node at Murdoch University. So this is another big project that about the horticulture genomics. So what I want to say that we have been using the genomics in different crops. This is either in the wheat or legumes and the horticulture crops. Now very quickly, if we deliver the varieties, does this mean that we can realize the full potential of genomics? Probably not. Why? And especially I'm talking in that developing countries where we are having the breeding program, public breeding programs, and then farmers, they need to be provided the seeds, etc. And for that, we came up with this concept that rapid delivery of the new cultivars to farmers is really required, because many times breeders, they develop great varieties, but they do not reach to the farmers. So our concept was that now the varieties, either they are coming through the traditional breeding or through genomics or even through GM or gene editing, we need to ensure that they are reaching to the farmers in the real time. So we need to strengthen the seed system. We need to think about that integration of those decision support tools in the farmer's hand, because now farmers, many of the farmers, they got the access to the mobile technology. I know that MSSRF, they are doing great work in this particular area in addition to many other areas. The other thing is that we need to provide the better agronomy to the farmers so that they can realize the full potential of those genetics which is present inside the seed. And not only that, countries like India and many other countries in Africa, we also got a lot of post-harvest losses, so we need to address that issue. When we talk about enhancing the farmers' income, we need to connect the farmers to the market. And for that, we need to think about the value addition as well. We need to bring these processing plants also next to the farm houses or so that not only the farmers, but even the consumers can be benefited. So we had this kind of concept, and we implemented this thing in one of those big projects from the Gates Foundation, Tropical Legume Project, which had the 3 different phases. I had a privilege to lead the 3rd phase and some part of the 2nd phase as a principal investigator, and that some project coordinator with me who worked, Chris Ojivo, Emmanuel Monio, and several other PIs, and some people from the Gates Foundation. Dr. Sadiq Abete was also program coordinator, and Dr. CLL Goda was the PI of earlier phase, 1st phase and the 2nd phase as well. So this was a big project, more than $67 million investment during that maybe more than 14 or 15 years. And this project scale was that in the 13 different countries in Africa, India, and also Bangladesh. And we had working on the 6 different crops in many countries. And what we did, that we addressed some of those issues what I was talking. One, that we were using the breeding, genomics, and developing those climate resilient variety. But second is that now that how breeders can continue to make these breeding program much more stronger, how we can continue to deliver more genetic gains. So this was the second approach. And through this one, we delivered several varieties, including this high on groundnut varieties, which I was talking earlier. And anyway, so there are a lot of stuff in the across the different crops. Third is, which is very important, building sustainable and inclusive seed systems. Unless you are having these sustainable seed systems, you cannot make you cannot realize the full potential of genomics. You cannot talk about the sustainable agri-food system unless you got these really good seed system. And this was really big work. And we worked with the different national programs, private companies or so. In total, while together we were able, this project facilitated to deliver 304 improved varieties, production of about 400,000 improved seeds, which could be grown in 4.4 million hectare area, and which produce about 4.9 million ton grains, which is the value of about 2.6 billion US dollar across the 30 different countries during the course of 10-12 years. Socioeconomics analysis indicate that these projects, they helped about 22 million farmers across those 13 countries. And now some quick studies. When we started the project, this is either Ethiopia, Uganda, Nigeria, Mali or India. If you see these different figures, during those 10 years or those phases, production has increased, number of the productivity has increased, economic value has increased, and majority of time, everything in different crops have been increased or so. And we documented the achievements. This is not that there were always success stories. There were lots of challenges, lots of failures as well. We documented them in the form of some books, some articles, and all these things. This is so that now the other crop community, other countries can learn the lesson and they can deliver these things. In fact, in terms of those impact of this study, and Gates Foundation, they recruited an independent agency, and this was from that Evans School Policy Analysis and Research from University of Washington, and they did the analysis. And then their analysis indicated the average benefit to cost ratio across segment is 16. This means that every dollar invested in TL, tropical legume, generated about $16 in value for a smallholder farmer. So this was that way that we were able to deliver through the genetics, through the breeding, through agronomy, through public policy support. And for this work, my previous organization, ICRISAT, and I feel very proud to contribute that we got this Africa Food Prize in 2021 for this amazing work across the 13 different countries. Anyway, friends, I wanted to highlight some of these success stories and would like to mention that the vision what Dr. Swaminathan had, that to ensure this nutrition and food security, which is a big thing, we all can contribute in this direction, and I think there are different ways. One is crop diversification is really essential for sustainable agriculture and ensuring food and nutrition security. When you talk about developing climate resilient, nutritious varieties, new approaches of breeding, including genomics, including GM, including gene editing, we need to use those kind of new approaches. We need to have the government support for taking all these products to the farmer's field. Nowadays, new approaches, including artificial intelligence, machine learning, and simulation approaches are expected to accelerate trait mapping, identify optimal parents, predict agronomic performance of breeding population. Countries like Australia, United States, Europe, this is not a big problem because here the breeding is basically commercial breeding and they know that how to replace the varieties, and farmers are also, they understand all these things. But now in the countries where we are having a smallholder farmers, we need to ensure that we need to have the rapid delivery system. Finally, international, national and local government agencies support and capacity building is a must for sustainable agriculture and food security. Finally, I would like to mention that as we contemplate the life and legacy of Professor Swaminathan through this M.S.Swaminathan Centenary lecture series, which is really fantastic and I feel really honored to be the part of that, let us carry forward the torch he ignited. Honoring his memory involves continuing the journey he initiated, a journey towards a world where no one sleeps hungry. He epitomized how science can act as a catalyst for positive change and showed that research can be directed towards addressing tangible challenges. His legacy continues to inspire researchers like me and policymakers to tackle the pressing challenges of our time, from climate change to sustainable agriculture. With these words, I would like to thank large number of collaborators who were involved in this work, what I presented, and now many of our partners here in Australia, because we are we feel very privileged to have this support and collaboration with large number of partners, and thanks to Murdoch University leadership, Grains Research and Development Corporation and their leadership, Hot Innovation, and many other organizations, they are generous supporter for our research. And of course, my team, which is having very young and diverse team, they are very hard-working people, including the senior researcher, postdocs, PhDs, and my colleagues from the laboratory, and so they are doing amazing work. Thanks to all of them for doing the great, fantastic work. And with this picture in 2015, when I was sitting in that feet of Professor Swaminathan, this is also one of my favorite picture, which we got in 2015 in ICRISAT. With these words, thank you very much, and back to you now, and we'll be happy to take up any question if there is a question. So.
So thank you very much, Professor Varshney, for beautifully summarizing how you can leverage collaborations, utilize varieties from gene banks, and use the power of genomics to deliver on products and varieties for agriculture. So also highlighting going beyond genomics, and also looking at how the adoption of these varieties can be a critical issue, and their utilization also. So we'll open up the session now for questions. So maybe we'll just go online and see if there's anybody who wants any questions. Please raise your hand or type in the chat box. In the meantime, if there's anybody in the audience here at MSSRF, we will begin with them.
Thank you very much, Rajeev, for that very excellent talk you gave. It was really beautiful. You talked about seed systems, and I think that is really very critical, I think, for any agricultural system, and particularly, I think, for small farmers, and to ensure that they improve their livelihoods. So what do you, in your experience, of course, you've used genomics a lot to improve these crop varieties, but as far as access to quality seeds is concerned, what are the major challenges and gap, particularly for smallholder farmers in Asia and Africa? And how does one build a system which does not exclude the good land races or traditional varieties, which have its own good properties, compared to some of the hybrid varieties that may be more commercially pushed and accessible to people?
Thank you very much, Soumya ma'am. This is a very good question and very important, or a key challenge in developing countries. So what is happening? So when we are having these improved varieties, farmers are not able to access these seeds in the real time. Point number one. Now, how to address that one? Though in countries like India, at the national level, at the state level, we got those national seed corporation, state seed corporation or so. What we need to do, we need to strengthen them. So as soon as we got the new varieties, they need to have a mechanism that they should be having very quick multiplication of the seeds. Second, the knowledge dissemination in the farming community is very important, convincing the farmers to grow these new varieties, because otherwise farmers, what they have been doing majority of time for self-pollinated crops, they take the seeds from the previous generation or from their friends or so, they keep on growing. And if you will tell them, because I know one example in Africa, for instance, farmers, they have been growing a variety and they call it Chinese variety. This is not a variety name, but some seeds came from China 30-40 years ago. Farmers, they continue to grow the same variety, and when I was visiting Ghana or Nigeria or so, when you talk to farmers, oh, this is the variety, and when I talk to those national programs, they say, well, we got the seeds, but they are not reaching to the farmers, because many times farmers say, we are very happy with this one. So in those cases, you need to create the awareness, and what we did in that case, we organized like farmers fair, and we say to farmers, don't worry, we will be growing our Chinese variety, but in addition to that one, we will be having some new varieties, and at the time of the harvest or so, we brought farmers from the different districts, different villages, and they can see that, well, which variety is really good or not. So then they say, yeah, this is really good variety, the new one. Then they ask from where I can get the seed. And now when you talk to the public system, they don't have the seeds. So then in that case, what we did, we worked with the private companies. This project was funded by the Gates Foundation, but they wanted to have the sustainable seed systems. But what they did initially, we put some money, we gave this thing to private companies, we also gave some incentives to that public seed system, and we were able to cater the need, and we fulfilled this demand of those farmers, and we were able to deliver those high quality seeds or high yielding variety seeds to the farmer. As a result, we were able to produce larger quantity of seeds. So there are some of these issues, and but now country like India, they are doing great job, especially from the seed replacement rate. But still, we need to see that what is happening in country like Australia or so, many times in wheat or so, when you are having these commercial breeding companies, you already know that after 3 years, after 5 years, we will be replacing this variety completely. This is not possible. This is not happening in those many developing countries. So we need to address those issues, and some of those lessons what we learned from those tropical legumes project, they can be implemented in many of those areas. Thank you for this good question. Yeah.
Thank you, Professor Varshney. My name is Jayaraman. I work on climate change in the foundation. Thank you for a very inspiring lecture, but a couple of questions. You mentioned the cost-benefit ratio, and which is of course very impressive, but what does it include? Does it include in particular costs of dissemination? And you know, and there would be a period before the varieties are fully established in the field as what farmers cultivate.
Yeah.
There would be a period of handholding. That's one part of the question. The second is, what is your idea from this kind of experience that you have for varietal development, which can take care of future needs, especially in the face of climate change? What kind of costs are we looking at in terms of adaptation? This is a big issue of adaptation costs, very difficult to get numbers on it. So what's your take on that?
No, thank you. I think this is a very important point, and maybe I may not have the answer for convincing answer, because this is a big issue. So this is not that just one or two point can address that one. So, but in our, to answer your first question, in terms of this cost benefit ratio, what they did, that they included the cost on the development and the dissemination versus that when you are getting the dollar value in terms of that harvest or that output. So this, they included all these costs. In this case, there is always a gestation period. So for instance, that if you start from today, then this will be taking some time for developing those varieties, and they will be reaching to the farmers, but then you need to have a pipeline. So for instance, and this pipeline should be a continuous process. So if you started now, and then today what you will see, the product, they are coming from the research what was done 3 years ago or 5 years ago. Now research what you are doing today, this will be delivered 3 years later or 5 years later. So this kind of pipeline we need to have. What happens majority of time, and this is one of the issue with the Gates Foundation as well, that Gates Foundation come up with the projects. When project is finished, then pipeline is stopped, and then maybe the new project will be coming, then they will need to start this pipeline again, but then you already got the gap. And then, so we need to address this issue, and this is not only with one funding organization, but this is the serious issue, unless government system or private player, they come in the picture. So I think that this is one of the things. Now second is, in terms of climate change, and again we understand these things, that you are having the issue of the mitigation, you are having the issue of the adaptation. So now crop improvement or breeding, they can address some issues but not all the issues. So for instance, you can develop some of the varieties which can provide the higher yield under that high temperature or higher yield, etc. But there are many other issues when you talk about the climate change, and agriculture is a broader aspect, and when you talk about the livestock, etc., those things are not coming. So anyway, so this mitigation, adaptation, there are two important things. Crop improvement can contribute something but not everything. So this is my take on this aspect. Thank you.
Thank you.
Thank you, Professor Varshney. I think there are a few questions in the chat box, and maybe we'll just address, put it up to you.
Yeah.
Yeah. So I think one question was, the question is, what has been the the adoption rate of these improved varieties by farmers? The question is by Rama Narayanan.
Yeah.
Yeah, so maybe you can just comment on that.
Yeah, so what happened that now, and some of my colleagues, I don't know that people from chickpea breeding right now they're in this group or not, but people like Bharat, for instance, he is working very hard in Madhya Pradesh and Uttar Pradesh and working with the different departments, and they are working right now in terms of enhancing the adoption of these varieties. So what is happening, and this is a very good point, so unless you are having some force not in the negative sense, but in the driving force, and then there should be some support, and if this is not the case, if you don't have this system, then the adoption will not be very high. And this is one of the key challenge in different programs. When I was working in that Africa with the Gates Foundation project, we were working in 13 different countries, and this, as they say, money acts like a glue, and this was like a driving force. We were able to enhance the adoption. When you don't have those things, sometimes, and as they say that batch value or something, that you deliver something, but then if you don't have this, so then this problem. But having said that, in the case of chickpea, we were able to have very high level of the adoption. This is still not reaching to the millions of the hectares, but thousands of the hectares or hundreds of thousands of the hectares this can reach, different varieties. And if you are working on chickpea, it will be good idea to talk to ACREP chickpea program. If you are working in groundnut, then they can also tell, because yeah, so this is really good point, and we need to keep working on this aspect.
Yeah. I think there's Rujuta Tayade, who's probably very, I think she wants to know how she can contact you. I suggest you do it personally through his email.
Yeah, so she
ID.
can send
Yes,
an email
yeah.
to me, and
So I
we can
think,
do that one,
yeah.
Mhm.
And I think somebody's just, Dr. Shankaran says, how best can we take care of land races and traditional varieties in various crops?
So what is happening? So this is very important, that we need to conserve the biodiversity, and this is really important. But somehow these land races, they don't have the higher yield, but they've got many important traits. That's what we have been doing, that now through the modern approaches, through the genome sequencing, through the pangenomics, super pangenomes, we can make the best use of those land races in the crop improvement program, and that's what we have been doing, and we are also doing now here in Australia. So this is really important.
Okay, thank you. And I think Raj Bhandari has just more like a, do you think native land races are more favorable to agroecological zones?
Yeah. So this is correct, that land races have evolved in time of withstand biotic and abiotic stresses. This is correct. But what happened during the breeding time, because you need to deliver more yield, people kept on selecting the genes or alleles for the yield, and they did not pay that much attention for those other alleles. But now, through the pangenomics approaches, we can bring them together. So this is correct.
Yeah, thank you. I think we have Professor K.C. Bansal who wants to ask you something. So, sir, can you please unmute yourself so that you can ask? Yes. So he's saying, yeah, please go ahead.
Yeah, well, thank you. I had anyway already typed my question in the chat box, but nevertheless, I wanted to actually congratulate Professor Rajeev Varshney for making this world-class presentation and kind of contributions he has made. You know, actually speaking, being a member of the MSSRF trustee board, I would like to congratulate him profusely, in fact, on my own behalf as well. And my question to you, Professor Rajeev, is that how can we, you have made a mark at the world level again on pangenomes, and you're talking of even more than 10,000, germplasm accessions of chickpea getting sequenced, and also you're into pangenomes of wheat as well. And there are very many traits which are very important, particularly thermotolerance in wheat or otherwise disease resistance in chickpea and others. But my question is, for example, now you have made a mark in genomics assisted breeding and pangenomes. How can we integrate genome assisted breeding or pangenomes with genome editing? which is another powerful tool, as you all know, for developing new, varieties or hybrids for that matter. Can we identify some of the deleterious alleles, from some of those germplasm collections or kind of pangenomes you have sequenced, and if we can delete those, SDN1 using genome editing, is that possible? Can we do that and further enhance the value you have generated, and bring it over even to the genome editing experts for developing more types?
Yeah.
Thank you. Yeah.
So thank you very much, Professor Bansal. You have been a great supporter and my well-wisher and mentor. So thank you for your kind words also and for all your support. And now coming back to your point, so yes, that's correct. And what we have done, and if you read, and you are also part of that paper of our Nature paper in this, which we published by sequencing of 3,000 lines in Nature. And the output of that paper is, many times breeders or geneticists, they have been always working towards accumulating the superior alleles and developing the great varieties. Now, through this genome sequencing, through that phenotyping data, we can identify, and we call this word genetic load. So genetic load means that in some varieties or in this different germplasm, you got deleterious allele in addition to have the superior alleles. Now you got two ways. One, accumulate superior alleles, or you can remove the deleterious alleles. And now you have the possibility either genomics assisted breeding, that you can purge those alleles, or you can use the genome editing. So now what we are trying to do, nowadays that's what we are going. So now from pangenome, thank you that you mentioned about pangenome, and we have done a lot of work in pangenome. Now we are going one step beyond from pangenome, and we call it cataloging of haplotypes. So what we are doing based on the genome sequencing, we are developing the haplotype catalogs. These haplotype catalogs, they are something like when you go to the supermarket, you go to the different aisles, you know where is the tea, where is toothbrush, where is this one, and which company, which quality. Same way in haplotype catalogs, based on the genome sequencing, germplasm sequence variation, we are trying to deliver those catalogs that this particular haplotype is superior for this trait, this is deleterious for this trait, and then this information is available with the breeding community, with the gene bank community, with the geneticist community, and now they can use this information through genome editing, and we call this approach called RAGE, R-A-G-E, removal of alleles through the gene editing. So basically that you are removing, or sometimes PAGE, promotion of alleles through gene editing for the positive one. So these are the good approaches, and we are working on those direction now. But thank you very much. And nowadays, this is the way that we are developing those haplotype catalogs across the different crops.
Thank you. Thank you very much, Professor Rajeev Varshney, once again.
I think, I think we have Mr. Dr. Narayanan also who wants to ask a question. So please go ahead. Yeah, you've been unmuted.
Yeah, can you hear me now? Narayan here.
Yes,
sir,
yes,
sir.
yes.
How are you? I can hear you very well. Good to see you.
Rajeev, so nice to meet you, and what a wonderful talk. I want to congratulate you for leading such wide-ranging collaborations and very successful ones, and for providing the leadership, technical leadership for such collaborations across geographies, across institutions. My question is, I also noted that there are not too many mid-sized private sector enterprises in that list of collaborators that you have provided. There may be a few of them, some may be foundations. But what would it make in your view to make the private sector step up onto the plate and then contribute, if not at the early stage, but at least at the dissemination stage of some of the products that may come out of such work?
Thank you very much, sir. Very good point. Maybe that when I was working in ICRISAT on the public breeding program, so at that time point we did not have many private sector partners. But now when we are working here in Australia, and in Australia for any crop when we work, even from the GRDC or from other organizations, this is impossible if we don't to get a project without involvement of the private partner. So for instance, if you see our wheat project, they are having the engagement of AGT, Australian Grains Technology, or Intergrain, or long reach plant breeders. If you are working in chickpea, we got Chickpea Breeding Australia, or the different companies. So what is, and this is one of the key things here in Australia, unlike many other countries, and I'm a big fan of this Australian R&D ecosystem. So what they are doing, when we work on any project, GRDC, which is a major investment agency, they would like to ensure that you are working with the private sector, because the breeding program is the commercial breeding program. So if they are not there, then our research outputs will not be, or there will not be guarantee that they will be uptaken by some company. But now when the company is the part of the program, they make 100% sure that these products or those outputs are translated to the breeding companies. So this is very good system in Australian R&D ecosystem. Now coming back to your question that how we can do this thing in countries like India or so, in Australia, even for wheat crop, which is self-pollinated crop, we are having the breeding companies, etc. When you talk company, including probably, and I know that you have great experience to lead several companies, to work with the different organizations. There are two challenges. One, that many times public system, they are always having little bit fear, or they are not very forward looking. They think that, well, if there is an issue of the trust with some people, not with all. So we need to address this issue of the trust, that if private sector are coming in the picture, this is not that they are coming just to make the money. In my opinion, unless you are having the private sector, you cannot make that kind of impact which you can make that if you are having the collaboration. Second, private sector also need to come in big way in their R&D investment. What they do majority of time, they say we are the company, but if you ask, I'm not talking generic, but majority of time, even the seed companies, they just take the seeds from the different organization, or even sometimes even without telling, multiply, sell that one. Ask the question, what kind of R&D investment they are making? So this needs to come from both sides, and I think this is really requirement of the time, and that's, so these are some, we can discuss more in details when I come to India. So.
Sure, Rajeev. I think you have touched upon the main points. Thank you very much. Thank you.
Yeah, thank you.
Yeah, so I think Kavya has a question. What are the challenges with interpreting right targets? Because the more we know, the more confused we are with the wealth of information. What would you suggest regarding the interpretation part? In a biological system, everything is interconnected. And where are we standing in the identification of key regulators?
This is very good point as well. So what is happening, and this is the problem. So sometimes they say, as they say, okay, that ignorance is bliss. So if you don't know that, probably this is good. If you know something, then you start to get confusion. But remember, knowledge is very, very important. Knowledge, we should be more and more curious. We should never stop. So this is my feeling, and I have been following on that one. Now coming back to the point, absolutely right. Even genes, they don't work in silo. Okay, I always keep telling my students, my postdoc, and my colleagues. Many times people say, hey, I identified the gene for drought tolerance. Question is, first is drought tolerance is not a simple trait that this can be controlled by just one gene. Even if you say 5 genes. Now what we do as a PhD student, as a postdoc, we work on our individual gene, we say this, this, this. But the question is, when you put these things together, you never know that there is synergistic effect, negative effect, how effect. So now what I am doing, and I have not put this thing in my presentation, we initiated these approaches. In ICRI said we also worked a lot on the different aspect. We talk about the systems biology, not the system approaches. System approaches are happening in the field when you are bringing the agronomy, breeding, physiology, all these things together at that higher level. I call it systems biology, that you try to address, even when if you want to identify the regulator. Question is, how many tissues, how many genotypes, what kind of approaches you have used? Did you just, when people say that I did the transcriptomics, I found so many genes upregulated, downregulated. But when you correlate these things with the proteomics, when you correlate it with other approaches, what number have increased down? So what I'm trying to say, you need to use the integrative biology approaches or systems biology approaches, and then you need to see that the genes which you were found in one experiment, how this worked in the other one, and how this integrates. And right now, that's what in some of our papers we mentioned, if you are not able to do everything by base, because it's not possible to do all these experimental setup, but nowadays machine learning approaches are so powerful that based on these machine learning approaches, you can define your important genes, and then after that you can validate them, and then you can use them in the crop improvement. So this is a bit complex, but this is very much required. Thank you, Kavya.
Yeah, and I think, yes, so that's about it. Are there any more questions? No. One more? Okay, Dushyant Singh Baghel. Hi, Dushyant. So I think your question is, thank you for the insightful presentation and congratulations on your remarkable contributions to agri-genomics through global collaborations. How can Indian agricultural researchers enhance their participation and visibility in international collaborations? I've often observed an underrepresentation of Indian genomics scientists in major global research initiatives. Incidentally, Dushyant is also a person who runs a genomics company, so I think his query is quite relevant. So please go ahead.
Yeah, Dushyant has been my old friend for a long time, so, and Dushyant knows my answer as well, but he wants to listen from me in this seminar, which is fine. So listen, I don't want to be the politically correct, but the one of the issue is, and this is, well, I moved to Australia now, but I worked it for 16 years in India, though in a research ecosystem, but I had the similar kind of challenges. You know what needs to happen, that there are several things. One is that many times when we work in the public system, we have this fear, many fears. One is that if I will discuss something, people will steal my idea. If I will share my data, they will publish these things. If I will go share my DNA, people think that even from the DNA you can create the plant, so that I should not share my DNA of this thing, or lots of issue. Or if I will have the collaboration, my director will say this thing, or my director general will say these things. Unless you are having all these fears, issues, etc., I don't think that things are going to happen. For getting any permission, now even for either that research project, proposal, funding, programs, you got all these issues. And I think this somehow, and I feel very proud to be Indian, but at the same time I feel that somehow these are the inherent issues in our Indian system and we need to address that one. So now when we talk, even I know that when I'm sitting here, I always would like to have many more collaborations with India, with Indian organizations. I would not like to generate thing, but majority of time, I cannot tell you that how much difficulty I have, even not just with that scientists, with their directors, with their director general, vice chancellor, and all these things. And many times even some of the MAUs, we never get these things after spending 2 years. Now on the other hand, if you see my collaboration with China, they are very, very successful. When you work in China, when you work in US, when you work in Europe, things happen at very swift pace. Now our leadership, we need to address this issue, and yeah, so I mean, well, there are a lot of stuff that which I can share, but probably this is not the right platform. Only thing which I will like to say, that we need to be proactive, and we need to get rid of with this fear, and we need to think that, well, I also have burned my fingers in some of the collaboration, but this did not stop me to collaborate. So maybe in 30% of collaboration I was failed. Some people, they ditched me, this is okay. But I feel very proud to have this successful collaboration, 70% collaboration successful, which is fine. I'm conscious that my all 100% collaboration may not be successful. If I think that all the collaborators will be nice people, probably not. But this does not mean that I should stop collaborating with the people. I don't know, Dushyant, whether you got that answer for your question, but I don't know that whether I can tell more.
Okay. So I think we'll take a last question. Alagarasan from ISA Tirupati. Your opinion on the use of AI and ML in GWAS, please. Have you tried any AI-assisted GWAS for any trait, and what are the challenges?
So thank you, and this is the new approach, and right now one of my PhD students, her name is Celia, she is working on this aspect together with Vanika and many other colleagues. So what we are doing, that we are using the approach. So now what happened, that in the case of 3,000 chickpea genomes, we got the genome sequencing data, we got the phenotyping data. Based on these things, we are developing some models. And once we got these models, what we will be doing, that we will be using them on 10,000 genome sequencing data. And based on these things, we will predict the phenotype based on the AI approaches. And then we will say, for instance, for the seed size, these 20 genotypes, these 50 genotypes, they are better. Then we will put those genotypes in the field, and we will validate our approaches. So these are very good approaches, and right now, people around the world, they are using these approaches, and we hope that we will be really successful in that thing.
So I think there may be many other questions, so maybe we can address them to him through email if you are really interested. So thank you very much. We bring a close to this session. If Professor Varshney's worked with 70% collaboration success and got so much, then you can imagine what he would have done. He was at 100% set. So he's been responsible for making genomics all the rage and bringing it onto a page now. So thank you very much for all your insights and your time, and all the audience present in MSSRF as well as online. Thank you for your participation and involvement in this lecture and in making this a success. Thank you very much, everybody.
And thank you once again for giving me this opportunity. I'm very much happy to see many senior colleagues sitting there, and especially Soumya ma'am, and also to you, Gayatri ma'am, and all the colleagues at MSSRF and all colleagues who joined this presentation. I really feel very honored for this invitation. Thank you once again.
Yeah, we'll make a copy of this available on YouTube to you, sir, later. Thank you. Yeah.
Thank you very much.
So we're all logging off now. Thank you very much, everybody in the online audience. Yeah.
Bye-bye.
Thank you. Thank you, Professor Rajeev.
Thank you, sir.